Okay then, let's have a look at the finalist presentations. Welcome to the stage, our first high school finalist, Basis China.
All right, so before our presentation, we have a short little story to present, and please enjoy it before it.
Thank you. And now there you are. Kid, what do you think you're doing, huh? Why are you sleeping in my class? Answer me.
Sir, I didn't sleep. I didn't sleep well last night.
Well, that sounds like a personal problem to me.
But sir.
No, shh.
It's not working.
That's enough.
It's cutting
You know what?
in and out.
You're currently having a
cutting
GPA
in and
of
out.
2.5, and nobody
Yeah,
can help
so,
you
like, was my
if you
microphone
keep
on the whole
doing it.
time?
Now you better catch up yourself and stop bothering me.
I have the beep and it's cutting in and out.
But sir. Sir, have you just not been a student before? Can you just have a little sympathy for us?
Okay.
Have
I
you
thought
ever
had that
been in a
other one that
state
worked. I
where
thought you
all
had that
lights
other
have
one.
gone off and you're the only one up trying to finish your homework? Have you ever felt the pressure of your parent peeking around in the corner of your
Oh, why
bedroom?
did it work yesterday?
Ever felt the eyes, ever feel the peers pointing holes into you? This feeling, you all just don't understand. No one do. Nobody understand.
But I do.
What?
I understand. It's stress. I've also once been in the same situation before. But now I'm out, and I believe it'll work the same for you.
Impossible. How? There's no way out of stress. There's just no escape.
Well, there is. And believe me, I can lead you out. Hi, we're Basis China 2023. We're a group of 18 high schoolers from 5 different Basis campuses across China. And we're here today to present you our project CyanoScent, which is, as its name suggests, producing scent with cyanobacteria. Now I'm going to brief you on a contest, which is already illustrated by a play. Stress is a state of worry or mental tension caused by demanding circumstances. Moderate level of stress can be beneficial to individuals by allowing them to have more motivation and focus more on their task. However, excessive stress can lead to a number of negative physical and mental health consequences. If an individual is unable to cope with stress, the physiological reaction will not moderate and will lead to a cumulative burden of chronic stress and regulatory systems, known as a high allostatic load. Constant stress experienced over a prolonged period of time can cause severe health issues. It can increase the risk of health problems like anxiety or depression. As reported by the WHO, 20% of children and adolescents worldwide suffer from mental health conditions. Also, chronic stress damages the body and the brain, resulting in non-communicable diseases, also known as NCDs. NCDs are the leading cause of death worldwide, killing 41 million per year, as reported by the WHO.
So through further research, we noticed that in comparison with the traditional methods of relying stress, aromatherapy is a better choice. However, the current methods of producing fragrance, which are chemical synthesis and natural extractions, are both unhealthy and harmful for the environment and human bodies. Firstly, the two main defects of chemical synthesis are the derive of petroleum byproduct and pollution. On the other side, the three main defects of natural extractions are the prolongated process of extraction, weak concentration of desired compounds, and lastly, a large waste of natural resources. Thus, by implementing synthetic biology, we believe that our solution is the best solution for this problem. Therefore, our project, CyanoScent, we aim to create a eco-friendly and self-sustainable product that can reduce pollution by introducing a brand new methods of production.
And to enable the cyanobacterial production of the terpene, we decide to introduce 10 different terpene synthase genes from plants into the cyanobacteria. And because our experimental process includes both E. coli and the cyanobacteria, we use the plasmids PMQAK1 as our shuttle vector because of its ability to replicate in various hosts. We also use the theophylline-dependent ribose switch, as well as the PTRC promoter in our design. The PTRC promoter is a stronger hybrid promoter, and this theophylline-dependent ribose switch will only allow the translation of the synthase when there is theophylline presented. We choose them because they are not normally presented in organism's biosystem and will therefore not likely to affect the normal metabolism of the organism. And by doing so, we are able to control the protein expression of the cyanobacteria and further the terpene expression.
In terms of a molecular synthesis, our team also takes efficiency and effectiveness into consideration. By using an already existing pathway within cyanobacteria, known as the MEP pathway, which synthesizes two chemical precursors to terpene molecules, known as GPP and FPP. GPP and FPP, similar to the monoterpene and sesquiterpenes, which are our desired products, are C10 and C15 molecules. Therefore, our team began to investigate into this MEP pathway and discuss the feasibility of using it to manufacture our desired compounds. And the investigation was proven to be a success. Through introducing plant-based plasmids into the genetic sequence for the MEP pathway, we were able to introduce a new enzyme known as terpene synthase behind the original 7 enzymes that already existed in the MEP pathway. Therefore, with a combination of the newly introduced plant plasmid, a highly efficient PTRC promoter, and the already existing MEP pathway that ensured the ease and stability of genetic editing, we were able to create an intricate engineered machine that effectively and efficiently synthesizes our needed fragrance molecules.
After collecting surveys on public preferences and conducting a lot of research and interviews to find out which fragrances can help us actually relax and calm down, we pinpointed six specific terpenes. These are pinene, limonene, bisabolene, farnesene, xanthine, and xantalol. To produce these terpenes, we created a cellular factory using CyanoScent, a monocyanobacteria at our chassis. However, as high school seniors ourselves, we initially started using E. coli as it's relatively easier to manipulate for us. We initially conducted our experiments using E. coli DH5 alpha and BL21 to construct our plasmids and to verify the expression of the proteins. However, to better achieve our goals and after consulting numerous experts, including Dr. Wang and Dr. Wei, we became certain that cyanobacteria would be a better suited chassis for our project as it better corresponds with our concepts of eco-friendliness and greenness. Therefore, we proceeded to transform our constructed plasmids into cyanobacteria CyanoScent through electroporation. And at last, we added dodecaine, which is an oil, into the cultivating culture to then extract samples to perform gas chromatography. We're able to successfully produce GC mass-detectable beta-pinene and alpha-farnesene in CyanoScent.
Elaborating on the terpenes, we have selected these six fragrances mainly due to preferences detected by our human practice and also various biological contexts. For example, in human practice, we have determined that people really like limonene, which we have increasingly attempted to synthesize multiple times which though we have failed, we will continue doing it after this project. Also others such as xanthine and xantalol, which come from the sandalwood tree, the ecological effects of this fragrance production have caused it to be driven to near extinction. And therefore we are trying to use cyanobacteria to nullify this problem and save the environment.
In order to allow our projects to reach a broader audience, we've developed bioreactors. Our initial experiment was conducted using fish bars and jars in different team members' homes to understand the impact of light, humidity, and temperature on wild-type cyanobacteria growth. And then bioreactors were designed to create a stable environment for cyanobacteria. And two types of bioreactors were further designed. Large ones resembling flowing water sceneries in public spaces, and small ones resembling daily items associated with fragrance. These designs aim to blend well into daily life and provide a soothing sense. And our project successfully improved our bioreactors, making them visually appealing, practical for daily use, and promoting a sense of happiness and greenness.
As for our modeling aspects, our team wanted to measure the OD values for our algae culture, and which was done consistently as a source of data for our team. An OD, or optical density value, is a measurement of a given wavelength of light through a translucent medium, which in other words measures the relative biomass of an algae culture. Normally, you would measure this using special lab equipment and regulated conditions which are unlikely to be found at home. So instead, our team devised a simple software that approximates an OD value given an image of algae or cyanobacteria growth. To do this, we extracted the RGB values from each image to then calculate the intensity, which is related to absorbance and thus the optical density of the material. Although this type of approximation method is slightly inferior to actual OD measurements, it is still within acceptable measures. This tool eventually proved to be extremely helpful in terms of quantifying our homegrown algae data, as well as taking quick measurements to test for our bioreactors.
Even though we've developed bioreactors, our project still concerns with microorganisms introduced with heterologous genes being brought out of our lab. Therefore, we've constructed a suicidal kill switch based on the type 2 toxin-antitoxin system, which will be turned on where there is limited iron ion present. While there is enough iron ion, our cyanobacteria will just live happily and be happy. However, where there is not enough iron ion, the toxin will be produced to an excess, which will lead to cell death. This system will ensure that none of any escaped cyanobacteria will be able to survive in natural environments. This is because in natural environments, microorganisms use iron ion because it is essential for their metabolisms, causing there to be a deficient of it. And out of the 4 plasmids we've constructed, 2 of them have been successful.
Okay, so besides investigating inside the laboratory room, our team have devised to expand our influence outside the experiment room for spreading the concept of synthetic biology and refining our project. We hold multiple presentations at different basic schools globally. During lectures, we elaborated the concept of synthetic biology and the impact of chronic stress and our solution to the problem of stress. These deliveries serve to introduce synthetic biology to the audience, reveal the danger of being stressed for a long time, and providing insights of how synthetic biology can be used as a solution to modern issues. Communication is also essential for enhancing our tests, since it facilitates the exchange of ideas between our group, other iGEM groups, and biology industrial pioneers. We have been attending and researching so many public activities that are related to synthetic biology and our subject, which is stress. A case in point can be Southern China Symposium, which we foster the exchange of advice and comments between different iGEM groups. Hence, we can embellish our research based on the advice they have given and increase the awareness of stress
among the public.
We collected 236 questionnaires in all, and it shows that 33% are feeling stressed frequently, and 10% are feeling stressed almost all the time. Also, there's 26% are having a bad quality of sleep. These results illustrate the prevalence of stress among teenagers, indicating the seriousness of the situation. To solve this problem, we interviewed several experts. Firstly, we interviewed Dr. Wong about the inside of fragrance industry, the obstacle of manufacturing, and how can smell affect human emotion. We observed that smell has a significant impact on humans' physiological state, but the current way of producing them are heavy and expensive. So it occurred to us that we can use synthetic biology as a means of production, which is less expensive and environmental friendly.
Okay, so in total, we reached out for over 550 participants and over 10 experts in our research. And this gave us a lot of opportunities to gain the insights and gain the preferences of the public. Okay, so as you can see here, the research process and the expert interviews are closely linked together since it directly confirmed the essential need for addressing the problem of stress, and it also confirmed the effectiveness of aromatherapy. It also decided the 6 terpenes that we're using in our wet lab experiments. And secondly, the communication and education process is also important, not only to the students and the audience, but moreover to us iGEMers, because we first promoted the communication as we have held and we have actively engaged in several online experiments and offline presentations. And we gave a lot of comments to other iGEM teams, while we also received a lot of comments from the other iGEMers that really helped with our project. And moreover, in addition, we also, as we have previously mentioned, we also conducted a lot of education programs and educational presentations all over China. And from the surveys that we collected, we can see that after attending to our educational program, a student has increased their knowledge on synthetic biology, on iGEM, on aromatherapy, and also on our project. So all these activities has greatly contributed and shaped the development of our project, and they have made us who we are standing here today.
In the future, we will continue to develop our project to the next level. We will continue to perform gas chromatography to test the other terpenes. We will also increase the production of the terpenes so it can be smelled by human olfactory system. We hope to make our hardware prototypes to life and truly achieve the purpose of relieving stress from people in an efficient, accessible, and eco-friendly fashion. Although we have yet to verify the effectiveness of our product in the community, we are certain that our project will work because of the research we did and the HP activities that we conducted. By cultivating cyanobacteria, we're developing a future where fragrance emanates from eco-conscious sources, thus promoting ecological equilibrium and reducing the strain on limited resources. Let's shape our future with CyanoScent. Spot greenness, smell happiness.
Thank you.
Thank you so much.